This is part 2 of chapter 31, picking up where we left off here. We're continuing to look at the Lophotrochozoans, and we've looked at roundworms and flatworms, phylum Annelida, phylum Platyhelminths. We're going to continue talking about phylum Mollusca, the mollusks. So these are, again, very highly diverse and monophyletic group of Lophotrochozoans. The characteristic body plan of these guys, they all have this structure called a foot. Pretty much they all have it. It's a large muscle used in movement. They have a visceral mass, which is going to be where many of the internal organs and an external gill are located. And they're going to have a structure called a mantle, called the mantle, which is an outgrowth of the body wall that covers the visceral mass and forms an enclosure called the mantle cavity. So we're going to look at these different structures and how they apply to the various types of mollusks. So lots and lots of diversification in mollusks. And once this molluscan body plan evolved, adaptations that allowed for better movement, feeding, and reproduction drove the changes that occurred in different types of mollusks. So different lineages, we've got the chitons, mollusks with dorsal shells made of plates. We've got bivalves, which are your clams and your mussels. Gastropods, your slugs and your snails, and then your cephalopods, your squid and your octopi. They all fall under the phylum Mollusca and have, based on their environment and the needs of the different individuals, have evolved vastly different adaptations to these three characteristic body plans, but retain some of the characteristics that make them mollusks. So let's look at the bivalves first, the clams. Most of clams' life is spent buried in the sand. We find them in both freshwater and marine environments. And we can see some of these various structures we talked about. We've got the foot here, this large muscular structure. We've got the mantle cavity down here, right? These guys do have a coelom in there as well, an opening. They have a shell, they have a mantle, okay? And all these things are inside of the clam. They're called bivalves because there's, an inward flow valve and then an outward flow valve where water kind of moves in, and the siphoning of that water is kind of where nutrients are obtained and passes over the gills and things like that. So I'm sure you've seen, clams if you've ever been to the beach, pick them up, dug in the sand, and were able to, look at some of these characteristics. Really amazing how they move hydrostatically with that, the muscular foot. Pretty amazing. So some of the key features, again, we've been talking about. The visceral mass separates the organs from a hydrostatic skeleton. So the skeleton is, going to cover the entire length of the structures, and hydrostatic means able to kind of move through the water easily. Something called a hemocell is a structure that separates the organs from the visceral mass. The muscular foot, like we talked about, serves to dig, move, feeding. You can see the muscular foot here, kind of pushing its way out of the side of the bivalve. The radiolata scrapes to obtain food in snails, clams, and filter feeds in the clams. And the mantle is what secretes the calcium carbonate shell. And depending on the type of species, whether we're talking about a clam, it's going to have two shells or one shell externally, and some have internal shells. Like with squid, they have a pen inside of them, which is their internal shell. And the production of those shells made of calcium carbonate is heavily influenced by the pH of the water. So we see that when there's ocean acidification taking place, these mollusks can no longer make their shells, and we lose them as part of the ecosystem, which can be potentially devastating. All right. So here's that radula that scrapes off pieces of food. You can see it zoomed in there, the kind of rough texture there, which allows this to, the snail in this case, to take in food. Many adaptations of the mantle allow it to do lots of things besides just secreting shells. So The bivalve structure, the two siphons in clams extend into the water column, control the in-current and excurrent of water over the gills. So that allows for movement and flow of water. In cephalopods, the mantle cavity fills with water. Mantle muscles contract, forcing a stream of water out of a single siphon. So you can imagine that provides a very strong outward flow of water and can allow for locomotion and propulsion of cephalopods, being our octopus and squid. Okay, so you can see that cavity opening up in the mantle, closing with water, forcing out through the siphon, and allowing the animal to move very quickly. So the squid can be gigantic or be very small, lots of different species, but here's the major characteristics of squid. They're bilaterally symmetrical. They have mantle, they have 8 arms and 2 tentacles. Shell is lost, but the internal support structure of the pen remains. They have these structures called chromatophores, which allow them to bioluminesce in a lot of cases, or change colors to match their surroundings. And, for squid that live in completely dark environments, having the ability to, ward off or to attract a lot of times prey, bioluminescence does that. They have gills. They move through propulsion, like we talked about. They have a structure called an ink sac. They have a complete digestive system. 3 hearts, 1 for the body, 2 for the gills. There's male and female. They can see, black and white, which is kind of interesting. They can taste and smell, but do not hear well. So if you've ever done a squid dissection, generally you can see many of these structures within the squid. For those of you that have taken the lab portion or will take the lab portion of this class at some point, you will do the squid dissection in the lab. All right, and here you can see, again, a photograph of the squid dissection and the various structures in there. So you can have this as a reference for when you're doing your homework or taking your exams. Okay, so let's move into the ecdysozoans. So these are the ones that, unlike the Lophotrochozoans, grow continuously, incrementally. The ecdysozoans grow intermediately, so they are going to molt. So that means they're going to essentially take their skin and they're going to shed their hard exoskeleton or the soft cuticle, depending on the type of organism, and they're going to grow into a new one. So the cuticle and the exoskeleton, the function of this, so how these things have evolved to have this structure, whether it's a shell or just an exoskeleton, it's going to protect the animal from predators if they're a prey species. It's going to provide for an effective structure for muscle attachment. Okay. During the times when an animal is molting, though, the animal is very vulnerable, and a lot of times predators can sense when that takes place. When the molting is taking place, and they will effectively go after these animals when they are molting, because they can, get to their insides and eat them much more easily than if they had their exoskeleton or cuticle. All right. So we can see here the Lophotrochozoans growing incrementally, and you can see on the clam that the lines represent that growth, that incremental growth. Whereas our ecdysozoans grow by molting. You can see this insect is leaving behind its old exoskeleton, and it will be growing a new exoskeleton. But during this time of transition, when it is molting, it is very vulnerable at this point to predators. Huge phylum within the ecdysozoa, and huge in terms of all animals, making up the largest percentage of that pie chart, is the arthropods. So arthropods fall under invertebrates, animals without a backbone. And what makes them kind of unique, they have, again, an ectoskeleton. They have jointed appendages. And 4 major lineages to look at: the arachnids, crustaceans, insects, and myriapods. So we'll take a look at those. So some of the key features of arthropods is that they are segmented. So the body is organized into regions. So for most insects, what you'll see is a head, a thorax, and an abdomen. spider or crayfish, they also have the cephalothorax and the abdomen, so they're just divided into the two sections. Exoskeleton made primarily of chitin. So chitin's a very strong polysaccharide, strengthened by calcium carbonate in crustaceans, which we talked about. And the ability to have these jointed appendages allows the much more movement, range of motion for these than if the appendages weren't jointed. Okay, so crustaceans being our shrimp, lobsters, barnacles, crabs, copepods. They are examples of ecdysozoans. They live primarily in aquatic environments. Okay, feeding appendages we're going to see compound eyes on stalks, and these guys are a very important food source. So crustaceans, pretty simple. You should know what a crustacean is. One of the hallmark crustaceans are the crayfish. These are freshwater crustaceans. They're like little lobsters. They breathe through feather-like gills. They have this hard plate in their back called a carapace. And we find them generally, in streams, feeding on dead and living plants and animals. Okay, so the basic body plans, they have structures that are involved in sensing the environment, the antenna. They have structures involved in defense and also, impressing mates as well, reproduction. Feeding structures in the mouth, walking, and swimming is towards the back with the tail. Right, and this kind of has the breakdown of what the different parts are in the crayfish and what they do. So I'm not too concerned about this information. All right, so where everything falls here in terms of our arthropods and the relatedness here, all of this very, very fun stuff down here, with the exception of horseshoe crabs. All of these are pretty terrible. Fall under a different family than the insects and the brine shrimp and the barnacles and shrimp and things like that. So that will close out this chapter 31 on the protostome animals. I'll see you next time when we will talk about the deuterostomes.